Okay. Hi. We've almost made it through the first week, guys. Yay. Exactly. So the plan for today is to finish talking about aspects of cells and organs of the immune system and then move into the humoral innate immune system, although obviously I'm a bit behind already somehow. But that's okay, we'll make it up sometime. So, in fact, yesterday you got to see many of these same blood cells that we were talking about on Wednesday. So you got to see the erythrocytes, you got to see, in some cases, some platelets, and you got to see a lot of leukocytes. And specifically, you saw some of the granulocytes, the monocytes, and the lymphocytes. On Wednesday, I kind of introduced you a little bit to the granulocytes and the monocytes together. The granulocytes and the monocytes together are both sort of the components of the innate immune system, while the lymphocytes, which I will introduce you to a little bit today, are the components of the adaptive immune system. So that's one big difference. One other term that comes up in a couple of places, so I'm just really telling you this to give you a definition of this word in case you see it elsewhere, is that granulocytes plus monocytes are sometimes called myeloid cells, while lymphocytes are the lymphoid cells. So if you see the term myeloid cells ever, it is an umbrella term for both granulocytes and monocytes. It's basically a general term for the immune cells of the innate immune system. And that really comes from this sort of developmental sort of pathway difference between these cells. We've got all of these cells that are developing from the same precursor. It's in fact the myeloid precursor. So all of those myeloid cells have this unique aspect of development, while the lymphocytes all come from the lymphoid precursor. And so, again, big part of this is just making sure that you are aware of that term in case you need it. And as I said, these cells, all the myeloid cells, are really the cells of the innate immune system, while the lymphoid cells are the cells of the adaptive immune system. So those lymphocytes can be subdivided into lots of different parts. So there are actually tons of different kinds of lymphocytes, though if you look at them by right stain, like you did in lab yesterday, you can't really tell the difference between any of those types. So you just call them lymphocytes, even though there are tons of different types. In fact, there are some times where I look at my immunology colleagues who are making up a new lymphocyte that acts against fungal infections in your left pinky fingernail on Tuesdays. And I'm like, guys, we might be dividing them up a little too much. So there are lots of kinds. They include things like T cells and B cells, which are B cells are the ones that are making your antibodies, as well as a few other kinds of sort of more specialized cells. And so here you can see we can actually divide up lymphocytes in a whole lot of ways. And we are going to learn so much more about lymphocytes when we talk about adaptive immunity. But now kind of this is the general idea that you want to know. One thing to also notice here is that the lymphocytes are the cells that live the longest of the cells of your immune system. So they are kind of second most frequent after the neutrophils, and they are very long-lived compared to most of the other cell types, which tend to be pretty short-lived cells. So you're kind of always making fresh versions of your myeloid cells, whereas your lymphocytes you're kind of making and keeping for a really long time. So that's one other big part of this that is a difference. We also need to talk a little bit about organs of the immune system, kind of anatomic locations that are important. Because that's one thing that's sort of different about the immune system compared to a lot of other body systems, is that other body systems you can often think about the place, the location, whereas the immune system doesn't have that kind of central place. You know, the cardiac system has the heart. The immune system is sort of more distributed around your body, so we need to think a little bit more broadly about the organs. There are lots of organs that have some kind of function in terms of the immune system. Just like with the cells, immunologists like to divide those organs up into groups. And so there are 3 groups of immune organs that we usually talk about. So the way that we group our immune organs is into primary, secondary, anybody want to guess the third one? Tertiary. You see, we're not very creative with our names. And so we're going to talk about both kind of how we define each of those 3 types and then which organs fall into those categories. This figure shows many of the critical primary and secondary lymphoid organs. In this figure, the primary lymphoid organs are in red, and the secondary lymphoid organs are in yellow. So to start with, we can talk a little bit about the primary lymphoid organs. And as you can see from this figure, like I just said, the primaries are in red here. You have 2 primary lymphoid organs. And the primary lymphoid organs are the bone marrow and the thymus. And so you can also see these in this figure from your textbook here. So the thymus and the bone marrow are the 2 primary lymphoid organs. The bone marrow is stuff inside your bones. And in fact, I will show you some bone marrow out of the mice next week. The thymus is another organ that you will be looking at in the mice next week. The thymus is one that sometimes people get slightly confused about. So the thymus is an organ that has 2 lobes. It kind of looks like this. They're kind of connected at the top, and they look like this. And it sits on top of the heart. So it's actually that orange organ right here that's sitting on top of the heart. You can see it sitting on top of the heart here. The thing that people sometimes get confused about is they also are aware of the thyroid. The thyroid is an endocrine organ, makes hormones, and it's up in your neck. So thyroid's up here, thymus down here. And so thymus is the one that we're going to care about, not so much thyroid. So the thymus and bone marrow, and this is in fact the definition of a primary lymphoid organ, are the places where your immune cells develop. So this is kind of where you make all of your immune cells. This is the organs where those cells come from. Both of these organs have sort of complex structures where there are lots of cells that are sort of feeding the developing immune cells to sort of help in their development. And so they both have kind of important structural features to help support the development of those developing cells. And so that's really what's going on with the thymus and the bone marrow, is that you're doing all of your development of all of your cells there for your all your white blood cells. You could also say that the thymus and the bone marrow are the locations of hematopoiesis. They're the places where you are doing all of this blood cell development. One thing that I will just mention really briefly is that that is actually only true when after you have been born. So the bone marrow in particular is really important for hematopoiesis sort of after you're born. However, in the embryo and in the fetus, hematopoiesis is taking place in slightly different organs. So early on, in particular, the fetal liver is really important. And so it's not until a little bit later in development that the bone marrow really sort of takes over in terms of hematopoiesis. So that's what's going on in the primary lymphoid organs. Primary lymphoid organs at this level are pretty easy to understand. When we actually talk about the details of how those cells develop, our life will get a little more complicated. But right now, the idea that you've got two places that are sort of sources of the cells. That's where the stem cells are. The cells differentiate. They grow up to become white blood cells. And then they, go, sort of like get loose and go to the rest of the body and do stuff. That part's pretty easy. So things get slightly more complicated. Actually, not at all, but you'll see, with secondary lymphoid organs. So secondary lymphoid organs are shown in this image in yellow. And there are a lot of secondary lymphoid organs. And I'm going to show you some views from your textbook a little bit later. Secondary lymphoid organs are some of the organs that immunologists spend the most of their time thinking about. These are kind of are really key. And I'm going to give you a definition of the secondary lymphoid organs now. But then I'm going to kind of give you a bigger explainer of what's going on. So I'm going to give you kind of the one-sentence version, and then we're going to kind of take a bigger look at some things. So secondary lymphoid organs are really where immune responses happen. So they are where those white blood cells meet up with microbes in order to start the immune response. This is particularly important for the adaptive immune system. So really, the secondary lymphoid organs in particular are where the adaptive immune responses start. Sometimes I think about the secondary lymphoid organs as where the party happens. It's really where all of our microbes and our, especially lymphocytes, meet in order to do stuff. And that was kind of the definition that I got as an undergrad, and it was fine. And then we, got a slightly more in-depth definition when I got to grad school, and I was sort of like, oh, like this makes so much more sense. Like there were some things that were like didn't really make a lot of sense before, and now it's like, oh, that's like really cool and obvious, and like why did no one explain it to me that way before? So I'm going to explain it to you that way too. And in order to kind of think a little bit about the secondary lymphoid organs and kind of what's going on with the secondary lymphoid organs, I have to take a second to talk to you about the circulatory system. This is not a super complex discussion of the circulatory system. In fact, in some ways, you can think of this as the immunologist view of the circulatory system. So here you can kind of see some idea of the circulatory system. You can see that we have a big vessel, the aorta, where blood is coming out from the heart. So you can imagine the heart right here. And then you can see that we have branching vessels that get smaller and smaller and smaller until we get to the capillaries. And then you can see that we get bigger again. But in fact, really the first half is sort of the important part here. And you can actually see like the changes in diameter, the changes in area, all that good stuff. But what we actually need to think about for a second here is what this really means in terms of a couple questions of physics. Don't stress. You already understand everything I'm going to tell you right now. You just don't know you understand it. You already understand it, though. Okay? So I want you to think about the heart right here. Okay? And I want you to think about the heart as a really big pump that pumps things with a whole lot of pressure. You have seen situations in your life where there are sources of liquid that push liquid at really high pressure. Imagine you turn on the garden hose in your backyard at full steam. Or imagine you have a fire hydrant going at full steam. You have, a hose on it. It's basically what we're seeing here. Big pump, hose. Okay? That's what we got. So now I want you to imagine that situation. And I'm going to ask you about a scenario. So imagine you hook up your garden hose, turn it on full blast, and then you squeeze it. And the reason why you squeeze it is you are actually making the diameter smaller, just like the diameter gets smaller here. What happens if you squeeze your garden hose? Not good. That is correct. Can we be more specific about some of the not good? You might have explosion because you're going to have pressure buildup as that liquid coming at high pressure tries to fit through a smaller hole. You could have water start spraying in different directions. You could have the water stop moving, right? Those are kind of things you can imagine. This all makes sense, right? We can totally see this. All right, now I want you to look around the room. Notice all of you are doing this right now, and none of you have exploded while we're sitting here. Right? There was no exploding going on. Yet, in this scenario, as it's sort of described in very simple terms, exploding should happen. Because we're taking high-pressure fluid and putting it through increasingly smaller diameter vessels. And so, in fact, there's this problem of pressure in Effector. You can see changes in blood flow and pressure and all that here. Really, you just imagine this change in pressure. And so, in fact, we have an adaptation that helps us deal with this pressure. And so the circulatory system has an adaptation to deal with this problem. This is really something that is going on with the arteries, where we're going from the really high pressure to low. So I'm really kind of talking about the left side of this. The way that your arteries are set up is that your arteries have semi-permeable walls. And so as the pressure is really high in the arteries, water is actually pushed out of the artery. And so that actually decreases the pressure because you lose some of the water. It balances out the pressure. So the water actually is spilling out of the arteries. That's actually a really good thing because that means that we can bathe our tissues in liquid and other sort of nutrients that we need. So it's really useful. I had always learned that by the time the blood gets to the capillary is so small that it's just one cell, going through there. And that's partly because everything else got pushed out. That wasn't the thing I originally understood. But in fact, all the water has been pushed out by that point. So you may have heard of something called interstitial fluid. This is interstitial fluid. Okay, so you can imagine this. We're pushing the water out of the arteries in order to deal with this pressure problem. But now we have a new problem. What's the new problem? Yeah. How do you get it back? If all we did was we pushed the water out of our arteries, then you would turn into Jabba the Hutt. You would just be a big blob with water everywhere. So somehow that water has to get back into the circulatory system. You've got to collect it from in between all the cells and dump it back into the circulatory system in order to make this whole thing work. And so the way that we do that is we actually have specializations in our body to help us collect that water and get it back into the circulatory system. That is called the lymphatic system. Sometimes you may have heard of lymph. Lymph is just that fluid that's around everywhere. Lymph is sort of that water and stuff that's around everywhere. So you have all of this fluid outside of your cells, outside of your organs, just around. We have all sorts of vessels throughout our body. They're open, so if any of that water flows by, the water is going to go in. There are valves, so the water can only move in one direction. It can only move in the direction we want it to go. It can't go backwards. So basically, we just have these open-ended vessels that collect up the water. As it moves, it can only go in one direction. There's no pump on this system. So if you want to move the liquid through the lymphatic system, the way you actually do that is body movement. And so body movement is basically pushing that water around so that it can get trapped into the lymphatic system. And so all of these vessels are known as our lymphatic vessels. And the lymphatics you can really think of as solving all of these water and pressure problems. But evolution was clever here. Because if you think about it, you've got all of this liquid from your whole body that is potentially going to contain microbes from your body or any other problematic substances in your body. And so from time to time, in a few different places around your body, you have little surveillance places where you check out, is there anything bad in this region of the body? Were there any microbes here? And those are really, that's the lymph node. And so basically you have all of these open-ended lymphatic vessels where cells, where sort of all sorts of other components will all come in and they will move, largely pushed by body movements, to get to those secondary lymphoid organs, which is where the lymphocytes hang out and sort of check out all of the things going through to see, is that a microbe? Is this bad? And so the major thing that people think about for secondary lymphoid organs are these lymph nodes. You have lymph nodes throughout your body, acting as surveillance systems throughout your body for some of these different cells. So generally what will happen is that you will have different lymph nodes that do surveillance on different regions of your body. And so the lymphocytes will kind of check those lymph nodes and say, oh, this region, there's something bad in that region. I got to go do some work in that region. Because you can imagine the alternative is that a lymphocyte starts up here at this cell in the upper corner of your head and is like, are you infected? Are you infected? Are you infected? Are you infected? And goes all the way to the one in the corner of your toe. And you can imagine that by the time it does that, it could have missed a lot of infections while it's going. But instead, it just has to look at the lymph nodes, say, is this region okay? Is this region okay? Is this region okay? Is this region okay? And it's a much faster process. So you've got these lymph nodes. They are each draining different regions of your body. So for example, the ones that are here are draining your sinuses and kind of upper parts of your head. The ones under your armpit are draining parts of your arm. The ones in your groin are draining the top half of your leg. You've got one behind your knee that's draining the bottom half of your leg. And so there's basically a lymph node for each region of your body that's doing that surveillance. And also, just so you know, in the time that I've been teaching at Drew, there have been new sets of lymph nodes discovered. So while this may seem like, old-fashioned, old-timey stuff, so we've actually realized this kind of shows the lymphatics stopping up here at the head. If any of you guys have ever done research with Dr. Knowles or been in Dr. Knowles' lab, there's this covering layer on the brain called the meninges that they peel off and throw away. Yeah, we've actually found that lymphatics go all the way through the meninges, and the meninges are completely full of lymph nodes. Is one of the newer sets that have been found. So that's basically what's going on with secondary lymphoid organs. So we have the heart. It's pumping liquid and cells. Those cells go to the lymph node. And then we have these lymphatics that are going to pick things up and dump back into the heart, so that by the time we get to the heart, we have all the liquid, all the cells back. And so we've got sort of both the arteries and veins carrying things, but also the lymphatics. If you've ever heard of the thoracic duct, the thoracic duct is actually the thing that dumps all of the lymph back into the heart as it's coming all around. So there are some other places where it can collect and come back in, but the thoracic duct is the main place where the lymph is going back into the heart. And so we might have a lymph node in a particular location. It is draining that region of the body. You can see this lymph node draining the foot. And then the lymphocytes will just check out that lymph node, say, oh, this region has a problem. Let me go check that out, instead of having to check out the entire body. And so the lymphocytes are really just going to go from secondary lymphoid organ to secondary lymphoid organ and check each of them to allow them to kind of check regions instead of having to search throughout all aspects of the whole body. And so this is how the secondary lymphoid organs initiate those immune responses, is that they are the collection place where the microbes are getting collected up out of that liquid. And so they're the place where the lymphocytes go and say, and look around for their microbe. And start the process and then go to the other potential organs. So the secondary lymphoid organs that I think about and that most immunologists think about the most frequently are the lymph nodes, which are going throughout the whole body, draining all sorts of areas of the body. There are lots of structures in the lymph node. So the lymph node does have a whole lot of specific structure. It does have a whole lot of specific areas for different cells to hang out in. But that's basically what a lymph node is. Other really important secondary lymphoid organs include the spleen. I like to think of the spleen as a giant lymph node. Structure-wise, a lot of what it has is very similar to the lymph nodes. It's just that it also has some additional parts that allow it to filter red blood cells. So it has sort of this red blood cell job, and it's a lymph node. The spleen is the lymph node, in quotes, in air quotes, that drains the blood. So it's checking the blood while the others are checking the lymph in different locations. And it's also sort of working on red blood cells. The spleen is also, when we do dissections, really easy. And so when an immunologist is getting an animal and they want some lymphocytes, the spleen is sort of the easy source. You guys are going to be getting the spleen from the mouse and getting some lymphocytes because it's like the easiest one compared to some of the lymph nodes, which are much harder to work with. So again, I think of spleens and lymph nodes as pretty interchangeable. There are also other places in your body where you have organized lymphatic tissue. I'm going to sort of mention two of the more famous ones, but there have been a number of situations where people have realized that perhaps there are more than just these two. So one that I want to tell you about is called the GALT, which stands for the gastrointestinal associated lymphoid tissue. So if you think about it, your GI tract is going to come in contact with a lot of microbes. And so you have specialized tissues in your GI tract that can act as secondary lymphoid organs to help you deal with some of those microbes coming in. There are also some lymph nodes in that general area. You'll see them in the mice next week. But one of the most famous parts of the gastrointestinal lymphoid tissue are these things called Peyer's patches. So this is actually a small intestine of a mouse. And you can see that there are these little white bumps on it. There are little white bumps on your small intestine too. And those are all little organized secondary lymphoid organ areas. They aren't sort of big enough to be a whole lymph node, but they're just sort of little specialized areas that help do some surveillance in your GI tract because there are going to be so many microbes there. And so we've got Peyer's patches as sort of our gastrointestinal associated lymphoid tissues. There are also, fun fact, the appendix is also secondary lymphoid tissue. So lots of people are like, the appendix has no function. No, it's a secondary lymphoid organ. It's basically gall in terms of acting as a place for lymphatics and lymphocytes. The other big place where we have some organized lymphoid tissue is sometimes discussed in different ways as either MALT, BALT, or NALT. MALT is mucosal. BALT, bronchioalveolar. NALT, nasal. The basic idea is that your respiratory system also has some of these organized locations of kind of secondary lymphoid tissue in the midst of the other organs. You may, for example, have heard of your tonsils. Tonsils are a big part of the mucosal associated lymphoid tissue. They are secondary lymphoid tissue as well. And so all of these are going to be helping do surveillance in your mouth, nose, and respiratory tract, because again, you can imagine what with the whole breathing, you're going to be getting in contact with a lot of pathogens in those locations. And so you have more specialized tissue there. Yeah?
So when people get their tonsils out for having, like, getting strep too much, why do they not get more sick?
So great question. I'm going to sort of hit two things there. First of all, why do you have to get them out from having strep too much? These are secondary lymphoid tissues. And as we're going to see as we get more into the adaptive immune response, one of the big things that happens in secondary lymphoid tissues is when your lymphocyte says, aha, I found it, like strep in that case, they divide and divide and divide and divide and divide. And that's why you have swollen lymph nodes, because the cells have undergone so many rounds of division, they're packed. Well, in the case of the tonsils, when they get super packed, they actually start to impair your respiratory system, and they have to come out. Fortunately, you have so many secondary lymphoid organs that they can sort of compensate for that. So you might be a little slower in making your response, but there are other organs that are going to take over drainage of that region. There are some situations where people, for example, also have to have their appendix out, obviously, or have their spleen taken out. And in all cases, there are enough other secondary lymphoid organs that they can kind of take over the surveillance. All right, so we've seen here the primary and secondary lymphoid organs. And the last thing that we have to talk about are the tertiary lymphoid organs. So I am going to give you a definition of tertiary lymphoid organs. And then what I want you to tell me some organs that meet that definition. Okay? So I'm going to tell you what a tertiary lymphoid organ is, and then you tell me some organs that fit. Okay? So a tertiary lymphoid organ is a place where an immune response happens. It is a part of your body where lymphocytes are actively, we'll say, trying to get rid of a microbe, trying to kill some infection. We're trying to make some kind of immune response. So the secondary lymphoid organ is like where they got started, where they got activated. The tertiary is where they go to kill whatever the invader is. So what are some organs that could be tertiary lymphoid organs? Justin. Okay, maybe your lymph node. Although we usually talk about that as a secondary, there are situations where it could be a tertiary too. Yeah, Jamie. The intestines. So maybe you have some intestinal infection. The cells have to go to the intestine. Sure. What else? How about skin? Because you can have a skin infection, right? How about kidney? You can have a kidney infection. Yeah.
Wouldn't it just be anywhere you can get an infection?
Yeah, it's anywhere you can get an infection, which is basically every single part of your body. So we usually only talk, like your skin is usually only talked about as a tertiary lymphoid organ when you actively have a skin infection. It's like the place where the infection is and where you're trying to clear the infection. Your kidney is a tertiary lymphoid organ when you have a kidney infection, and on and on and on. But honestly, it could be any organ for a tertiary. So that's why they can't really color code it on this figure, because you would just color code the whole thing. All right, so that's what I have to say about sort of general cells and organs. So now we kind of know who's who. And I want to tell you a little bit now about sort of some of these details of the layers of immune responses. And so for the rest of today and all of next week, we're going to be talking specifically about details of innate immune responses and some of the specific things that go on in the innate immune response. Again, like everything else we've seen today, and sort of we've thought about with immune systems, we can, again, try to divide up the innate immune system. We can actually divide up the immune system in general. I've shown you cells. I've shown you organs. The other division that sometimes people will make is that sometimes there are proteins that are doing jobs versus cells. And so we can kind of think about active proteins versus active cells. And everything I'm going to be telling you about really for the rest of the day today, as you will see this, are about proteins in the innate immune system. So active innate immune proteins. Usually, those innate immune proteins are going to be really active against extracellular pathogens. So pathogens that are outside of cells. So very frequent, because we're going to have these proteins that are good at killing stuff. You'll see later sometimes where they are inside of cells to kill stuff, but you would imagine you'd want to put them outside of cells to kill stuff outside of your cells before the stuff even gets into the cell. So we're not going to see these being as important against intracellular pathogens like viruses, but all of these extracellular pathogens, when they are outside of cells, are subject to the actions of these innate immune proteins. Also, the other thing that's huge about these innate immune proteins is that they are effective right away. You have these innate immune proteins in you ready to go right now. They don't take any time to get prepped. And so they are going to start acting like 1 minute after infection. In fact, we really talk about the fact that they are what you have going for you for about the first 4 hours after infection. So 0 to 4 hours, this is all you got. Everything else takes a little bit of time to rev up. So these are super important because they're so immediate. You can imagine when we talked about the replication rate problem, that having something to immediately start pushing down numbers is going to be really important. And these are going to be important for extracellular pathogens. In one way that our different layers of the immune system kind of bleed together, if we look at many of the organs that we think of as barrier organs, most of them make a lot of innate immune proteins too. So they make these proteins often referred to as antimicrobial peptides. So your skin has a whole lot of antimicrobial peptides on it. Your stomach is full of a whole bunch of antimicrobial peptides, as are your intestines. Your lung contains a number of antimicrobial peptides. Your urinary tract contains a lot of antimicrobial peptides. And so you've got these antimicrobial peptides in a lot of the barrier organs. And so that's one place where it's hard to say, like, what's barrier defense and what's innate? And so these are going to be helping you out in a lot of different ways. There are tons of types of antimicrobial peptides. This is the table that your textbook gives you. The point of me showing you this table is to show you that there are a lot of them. I'm going to give you some specific examples, but I want you to realize I'm only giving you a couple of specific examples, only kind of hitting highlights. There's actually a lot more here than what I'm telling you. Just because it's cool, I'll tell you that one that's really cool that I'm not going to have time to really talk about otherwise is lactoferrin. So microbes need iron as part of their life, just like you need iron. Lactoferrin basically just binds all your iron and hides it, so microbes can't find it. So it's an antimicrobial peptide, and that's kind of its fun, cool activity. So some of these are acting in all sorts of really interesting ways. So one of the most famous ones that most immunologists, think about a lot is one called Lysozyme. So Lysozyme is the first one I'm going to tell you about. Just be aware, Lysozyme, which you can see up here, and you will also see the word on the next slide, is not the same word as lysosome, which is an organelle in cells. So we're talking about lysozyme. It's an enzyme. It has zyme at the end of its name. Lysozyme, I'll show you what it does on the next slide. But in fact, lysozyme is found pretty much in all of your mucosal secretions. So your saliva is full of lysozyme. Any other mucosal secretions are full of lysozyme. Lysozyme is actually at very high concentration in tears. Because it's in your eye and in your conjunctiva. So basically, any mucosal secretion is going to be full of lysozyme. And the action of lysozyme is described here. So very brief, most bacteria have a cell wall. There are some famous types of cell wall, like gram-negative and gram-positive. Most of those cell walls are made of this, have at least some of this molecule called peptidoglycan. The details of that vary from microbe to microbe. But everybody usually has at least a little peptidoglycan, part of their cell wall. Peptidoglycan is only made by bacterial cells. Your cells do not make peptidoglycan. They don't have peptidoglycan as part of their cells, as part of their membrane. And lysozyme is an enzyme that specifically breaks a chemical bond within peptidoglycan. It is depicted here as Pac-Man. And it specifically breaks a chemical bond in peptidoglycan so that the cell wall is broken in bacteria, their lipid bilayer is exposed, and those bacteria can be lysed. And so you've got tons of this. It's going to be great because it is only going to be able to act against bacterial cells. It can't hurt your cell at all because you don't have peptidoglycan. It specifically just works by breaking down peptidoglycan and making these bacteria susceptible to other types of attack. And so what you can see is that lysozyme is pretty good in that it is going to be able to affect most all bacterial cells, but not affect your cells. It's really what we want here. Yay. That is not the way that all antimicrobial peptides work. This is just an example to show you that that's not how all antimicrobial peptides work. I'm not going to talk about the specific peptides that are going on here. Also, I'm very glad that this is not when Dr. Miller was walking by, because I always feel like if she saw this experiment, she would freak. Don't do this experiment in Dr. Miller's presence. So what is happening in this experiment is that we have people who are actually taking bacteria and putting them right on their hands. So you can see that we have a person who puts Staph aureus right on their hand, or a person who puts E. coli right on their hands. Staph aureus is gram-positive, E. coli is gram-negative. And then they hang out. Just don't touch anything and let the bacteria sit there on their hands for 30 minutes. And then they put those hands on plates, on culture plates, to see if either the Staph aureus or the E. coli survive. And what you will notice is that the Staph aureus survives really well and grows. You can see little fingerprints. But the E. coli does not. And the moral of this story is that there are antimicrobial peptides on your skin that can actually distinguish between gram-positives and gram-negatives, and that kill the gram-negatives, but not necessarily the gram-positives. So I wanted you to realize that Lysozyme is a great example of one of our antimicrobial peptides that hits all kinds of microbes. But there are other kinds of antimicrobial peptides, like some of the ones that are on your skin, that are going to be more selective here and are only going to hit some microbes and not others. That's what we're trying to get from this experiment. And also, don't tell Dr. Miller that I told you to put bacteria on your hands. Because note that you have to do this on your hands, not on gloves, so that you can get the peptides from your hands to do the work. So the other really famous type of antimicrobial peptides that exist are known as defensins. So there are a bunch of different types of defensins. There are alpha and beta, and then there's all sorts of sort of subtypes of defensins. Again, I'm just talking about defensins as a group, though there are lots of individual members. What you should notice is that the different defensins are made by different kinds of cells and act in different anatomic locations. So that's really the difference between some of the different ones. You can see that many of them are acting in, say, the GI tract. That's a pretty big place. That's where we think about defensins a lot. In fact, really frequently when we think about defensins, this is an example of the GI tract, like the intestine, where you have those extended villi to do absorption in the intestine. And down in between them, you have this little area called a crypt. The cells at the bottom of that crypt are great at making defensins. And so they're kind of making a lot of defensins to get rid of any microbes that might come around. Also notice, this tells you about regulation of the synthesis of these defensins, and that most of them are listed as constitutive. And that means that your body's making them 24/7 to get rid of any microbe that might come around ASAP. One other thing that I did not mention that I should have, when we think about innate immunity, innate immune responses are responses that we actually see pretty broadly evolutionarily. So I told you before that this semester we're really talking about vertebrate immune responses. But innate immune responses, especially antimicrobial peptides, are found pretty much in all animals. There's probably some in plants. They're found really broadly. And there are some organisms where their whole immune system is just defensins. And I know a surprising number of chemists who work on defensins and don't realize they're doing immunology. And I'm like, really, guys? So we can also talk about how exactly defensins will act against microbes, to act on different cells. So that is shown on the left. I will tell you that when I first learned about defensins, I kind of learned this. Again, that was cool. And I was confused about it for years because there was a fact I didn't know. So I want to tell you that fact before I explain the details about defensins. So here you can actually see some lipid bilayers. You learned about lipid bilayers in Bio 250. You learned about the fact that there are these head groups and these fatty acid tails. And you can have different kinds of head groups on your phospholipids. Some of them have charges, like some of them are negative. Some of them are neutral. It turns out your cells are really organized with their phospholipids. They're so organized with their phospholipids, they put all the ones with a negative charge on the inside wall. There's actually a lot of reasons why that works. It's really helpful for things like electrical conductance, But it's important for a bunch of things. So your cells, all of your cells, put all the negatives inside because they're really good at organizing. I'm sure Donna already told you about flipase. That's the job of flipase. Microbes, particularly bacteria, don't do that organizing. They have negative charged phospholipid heads all over the place. So you can see eukaryotic, prokaryotic cell membrane. Defensins are just positively charged peptides that can poke holes in membranes. And so basically what you can see is that defensins are just these positively charged peptides that jam into membranes and make holes and destroy those membranes. And what I didn't understand for a long time is, well, why don't they destroy our membranes? The answer is because we hide all of our negative charges, and bacteria don't. So the defensins can interact with those negative charges on the bacterial cell membrane, but not on ours. And so that's actually how the defensins can act against a bacterial cell while leaving our cells alone. There are some other ways that defensins can act, but we're not going to deal with them now. This is just sort of classic defensins. In one previous year where I taught immunology, at one point in the middle of the semester, one of my students was like, I am super confused about something. And people often get confused. I'm not going to say it. This is like the only time in life that someone was confused. But someone was confused, and the thing they were confused about was that we were learning so many different aspects of immune responses. They're sort of like, well, why do we still get sick then? Like, look at all these things we got, and we'll still get sick sometimes. And so I just want to point out, and I sort of have these scattered throughout the semester, there are ways that some microbes actually try to get around some of these things. And so antimicrobial peptides are one of many examples where microbes do actually try to avoid that immune response. We have this sort of evolutionary battle where we're both trying to like get the upper hand. And so there are some microbes that actually break antimicrobial peptides, some that actually like try to export them, some that will coat them, some that mess with their charges. So the moral of the story is antimicrobial peptides cannot do everything. We must have more immune responses. So if another immunologist looked at my syllabus and looked at the title of what we're talking about right now of humoral innate immunity, humoral is usually a word immunologists talk about when they mean proteins that do stuff. There would be one thing they would think of. They would be like, oh, I know what she's talking about today. Because there's one other super famous aspect of proteins of the innate immune system. I'm not going to get through all of it today. It is, in fact, something that is many immunologists' least favorite topic. But it's really important, and so I want to talk a little bit about it. And in fact, I will tell you, when I was in grad school, we would basically like have a contest of who got the short straw and had to actually do the lecture about this topic because we were like, oh, really? We have to think about it. Because everybody, and one of the reasons is because when we thought about it as something that was really old-timey. Like it was a thing that was described in like the early 1900s, and it's like old and dusty in the textbook, and no one actually like cares about it now. This is something known as the complement system. And one big thing that has happened, that we have seen more and more often in immunology, is that we are reading, or we're realizing that complement can do a lot of things that we never knew about before. So complement has now been shown to be one of the major things leading to issues in arthritis. Both osteo and rheumatoid arthritis are very much complement driven. There's actually some role for complement in a number of neurodegenerative diseases. There's a role for complement in if there are all sorts of diseases that nobody used to think had anything to do with the immune system, and now suddenly we're realizing that they're actually complement issues. What you can actually see here is an example of someone who has a particular type of macular degeneration. This is an eye. And you see how there's all these little blobs in the eye? That's all complement deposits that are actually hurting this person's vision. And so complement is suddenly one of those things that we realize is more and more and more interesting and important for immunologists to know about. So complement comes off as one of those things that this is also something that students sometimes see and like freak out about complement. I think it's terribly, terribly complicated. It's really not, I promise you. And so so this is kind of a general theme of something we're going to see over and over as we think about complement. So we're talking about complement. Sometimes it is abbreviated as C prime. So if you are reading a paper and you see something about C prime, they mean complement. Complement involves a whole bunch of proteins of the innate immune system. You can see one example of such protein here called C3. These proteins get cleaved by some protease. And when they get cleaved into two parts, a big part and a small part. Usually the small part gets A, the big part gets B. One of those parts gets stuck to something. One of those parts floats away. Sometimes we don't even care about the part that floats away. We never see it again. But the idea is that a piece of that complement protein gets attached to something, usually to a bacteria, you can imagine. And so you can see a complement protein being attached to a bacteria here. Sometimes we refer to this as the fixation of complement. It basically means complement got attached to something. And so through all of the like craziness of complement, basically what we're seeing is we're seeing some protein get cut by some protease and then get stuck to something. And usually when it's stuck to something like this, it has a job. Spoiler alert. Its job is very often to be a protease, to cut a different complement protein. That's it. That's all we got. If you have looked at the blood clotting cascade, complement cascade is very similar to a blood clotting cascade. And so that's sort of relatively common in terms of what people have seen. So this is your textbook's figure of complement. Again, I want to give you a sort of general idea of what goes on with complement. Before we get to the weeds. So the thing I really like about this figure is that it sort of separates complement into three different areas. The initiation part, the amplification part, the termination part. I'm going to draw it even more simply first. And the way that I'm going to first draw it is tell you that there are three different ways you start the complement pathway. That's what they're calling initiation up there. So there are three different ways you can start. But if you notice from how I drew this, they all end up at the same place. So we've got three ways to initiate. Then they all get you to the same spot. That's what this is calling amplification. Then there are different ways that we can get rid of the microbe or do something. And there are multiple different ways we can get rid of the microbe. I'm going to introduce you to a term because honestly, I want to introduce it as early as possible in the semester because I use it all the time, like in my life, and people are always like, that's not a normal people term, that's an immunologist term. So I know I'm going to slip up and start saying it, so I need to tell you what it is ASAP. These are the complement effector functions. Effector functions are how you do something in the immune system, how you get rid of a pathogen, how you have an effect. So basically, we have 3 ways to initiate. We come together. We've got a few effector functions, 3-ish. This is basically what complement looks like. And so what happens is you can have whatever, you could initiate through one way, and then you could deal with the pathogen through one way, or you could go like through this and then come on this one, or then you could go through this and come on this one. And so the key thing that you need to know as we talk about these is, are you up here? Are you down here? Are you in the middle? And kind of like, which branch are you on? So people get really hung up with like memorizing all the names of all sorts of things. And like, really what you need to know is, which branch is that thing on? Like, yes, I can recite the order of all of them, because I've been doing this for a while. But really think about, what branch is it on? What does it mean if it's on that branch? Okay? So the way I have this set up with the rest of the slides is that basically what I do is I talk you through one way of starting, one way of initiating, and getting to this middle part. And so what I'm going to sort of start to talk about is this. Maybe I'll get through it today, not totally sure. Then what we do afterwards is we talk about how you actually deal with it, so the bottom parts, and then we'll come back and say, you could also start in these other ways, and I'll be like, oh look, now we're at a place you already know. Okay, so that's how we're going to do this. We're going to try to do this part today. Hopefully we'll get through it, may not. So the way that we, the first really described way of initiating complement is through something called the classical pathway. We now know that the classical pathway probably actually evolved last. It's sort of the most advanced in some ways. And this figure, actually, or this slide, tells you perhaps the most important thing about the classical pathway, which is that the classical pathway is initiated when an antibody binds to the surface of a pathogen. You can see antibodies here drawn as sort of little Y-shaped molecules. We'll talk way more about antibody structure later. Now you can think of them as little Y-shaped molecules. My friend's 3-year-old daughter has decided the letter Y is an antibody. Clearly my work is done. And so you can see that the antibody has an arm that can bind to the microbe and has a little tail of the Y that sticks up. An antibody binding to that surface is what starts the classical pathway. Reason why this is really important is that antibodies are part of the adaptive immune system. And so in fact, here's a way that we're seeing the adaptive and the innate immune system working together already. So again, we like to describe these as separate layers of the immune responses, but the classical pathway is one place where the innate and adaptive responses are working together. Once we have the antibody bound to the pathogen surface with those little tails sticking up in the air, the first of our complement proteins can act. And it will bind and recognize those tails up in the air. In fact, it actually recognizes some specifics of the geometry of them. And so some antibodies are better than others at doing this because of their geometry. That protein that acts first is called C1. Real hard here, real complicated. It's the first one. Complement starts with C, C1. This is actually what C1 really looks like. In cartoon form, here you can kind of start to see it. This is a little washed out as an electron microscopy version. It looks like an upside down bouquet of flowers. It really looks like an upside down bouquet of flowers. And that upside down bouquet of flowers, complement C1, binds to those antibody tails. that are on the surface of the pathogen. And this gives us active C1. C1 basically is only able to do things when it has bound to the antibody molecule on the pathogen surface. And when it does that, it starts the next part of the complement cascade, which I will tell you about more on Wednesday next week. I hope you have a great Labor Day weekend. And we will finish talking about this and start our way into other parts of innate immunity on Wednesday.